All right, I think we'll get started. Welcome, everyone. I'm Andy Aguirre. I'm a medical oncologist and a physician scientist here at Dana-Farber. I have the honor of introducing today's speaker, Dr. Elizabeth Jaffe, who will give this year's Krakoff Family Lecture. I'm sorry that we couldn't have this important lecture in person, but I think next year we'll plan to see everyone in person. First, I'd like to say a few things about the Krakoff family. Sandra Krakoff has been a Dana-Farber Cancer Institute trustee since 1995. In 2010, Sandra and her family, along with the Kuhlman family, endowed the Krakoff Family Lecture in memory of Anna Maria Kuhlman, who was the mother-in-law of Sandra's daughter, who unfortunately succumbed to pancreatic cancer the same year in 2010. This annual lecture was established to share advances in the field of pancreatic cancer by bringing a talented researcher or clinician to Dana-Farber to really showcase the latest in scientific discoveries and treatments for patients. So we're very grateful to Sandra and the Krakoff and Kuhlman families for all they've done for Dana-Farber over the years, for endowing this lecture. And we're really honored to hold this annual seminar in memory of Anna Maria Kuhlman. So today we're really pleased to have Dr. Elizabeth Jaffe, who's our speaker for the Krakoff Lecture. Dr. Jaffe is a professor of oncology at the Johns Hopkins University School of Medicine, where she trained in oncology and has remained for the duration of her career. And she holds a number of very important leadership positions at Johns Hopkins. She's the co-director of the Skip Raw Center for Pancreatic Cancer Research and Patient Care. She's the deputy director of the Sidney Kimmel Comprehensive Cancer Center, and she's the associate director of the Bloomberg Kimmel Immunotherapy Institute. She also holds numerous national and international leadership positions within the cancer research community. She's the past president of the AACR. She's been a scientific advisor to many, if not most of the most prominent cancer centers in the country. She's been co-chair of the NCI Blue Ribbon Panel for the Moonshot Initiative, chair of the National Cancer Advisory Board for the NCI, and she's currently a member of the board of directors for the Biden Cancer Initiative. And lastly, she's also chief medical advisor to the Lustgarten Foundation, through which she directs some of the most exciting trials and clinical research programs being conducted in pancreatic cancer research. And in addition to all this, Dr. Jaffe is a world-renowned medical oncologist and cancer immunologist. She's led a highly successful cancer immunology research lab for many years, studying the mechanisms of anti-tumor immunity, as well as anti-cancer vaccines. She's published hundreds of papers, written numerous patents, and I think most importantly, she's really a beloved mentor to many students, trainees, and faculty members throughout the world. So she, better than anyone, understands the challenges of treating pancreatic cancer and the critical steps that we need to take to improve immunotherapy response in this disease. And we're really honored to have her talk today, and she's going to tell us about turning up the heat on pancreatic cancer, lessons on overcoming a cold immunologic microenvironment. So Dr. Jaffe, thanks a lot for joining us.
Thank you very much, Andy, for that wonderful introduction. I'll just get my slides up. And I also want to thank the Krakoff family for funding this very important seminar series. So I'm going to be talking, as you mentioned, about pancreatic cancer. These are my disclosures. Happy to discuss any of them after the talk if you have any concerns. So I like to start off my talk with sort of a perspective in our field of immunotherapy. We've really made great progress over the past decade, starting in the first, with the first approval, FDA approval of an immune oncology agent in 2011, these immune checkpoint agents, but it's really based on 30 years of scientific discoveries that has brought us to this time of accelerated FDA approvals for therapies in immunotherapy. And the reason I like to point it out, I know this is a complicated timeline, but what this really is showing you is that the discoveries that occurred in our field really paralleled the discoveries of technology. And this is really the theme of my talk, technology. As the technology improved from PCR, which allowed us to start to sequence genes or the expression of genes, to sequencing the first human genome in 2000, to now currently being able to do these single cell analyses with next-gen sequencing, RNA-seq, and multiplex proteomics, we've made a lot of progress in the field, and it's really what's going to propel us to make more progress, not only in immunotherapy, but in other areas of cancer therapy as well. And so where is immunotherapy now? So immune checkpoint therapy clearly has resulted not only in some good responses, but in some real durable long-term survivors. And this is showing you the data on melanoma. And basically what you're looking at on the top is the overall survival, and then on the bottom is progression-free survival. And the important point here is that with melanoma, which is one of the tumors that responds very well to anti-PD-1 and anti-CTLA-4, we have a 5-year survival with one agent at 26% or even 44% with anti-PD-1. And when you add in the two together, you have almost 52% 5-year survival. This is really a big advance in our field, and obviously melanoma is a horrible disease. So to turn this disease from a death sentence to something patients can live with is really a wonderful achievement. Of course, we want to do this for many cancers, and that's what I hope to be able to show you some progress in pancreatic cancer today. We're also at a time of precision immunotherapy. We talk a lot about precision medicine. Well, immunotherapy has really become part of precision medicine. And our group was the first to show that it's possible to identify tumors by their genetic makeup rather than just by where they derive, what tissue they derive from. And we found that in particular, cancers that share a high microsatellite instability or high mutational burden are the cancers that will most likely respond to anti-PD-1 or anti-CTLA-4, some of the other agents that we have. And in fact, we were able to get anti-PD-1 approved very rapidly for this particular cancer because these cancers, even at very metastatic stages, respond very well to our checkpoint agents. But this has also opened up other areas. For instance, we're now able to take a small biopsy from a metastatic tumor and within 1 to 2 weeks, sequence the mutations and develop things such as vaccines for trying to induce a T-cell response against neoantigens in cases where patients' neoantigens are hiding from the T cells under normal circumstances. We're also able to sequence the T cell receptors from a specific patient's tumor for an antigen and develop these patient-specific cellular engineered therapies. So we've really gone in just such a short period of time from thinking about cancers as a group of diseases that originate from an organ to a genetically defined type of cancer and ones that can be we can develop personalized therapy. And so immune checkpoint inhibitors have shown unprecedented responses against a number of advanced cancers. So what do we know about those cancers that respond? Well, I think we all know that high tumor mutational burden cancers, such as single nucleotide variants, do respond to these tumors. However, we also know that there are other tumor antigens. And again, it's the new technologies that allow us to identify these other antigens. For instance, viral antigens in Merkel cell are very important immunogens. In renal cell carcinoma, there aren't lots of neoantigens, but there are very immunogenic neoantigens called insertions and deletions, or indels. And then even fusion proteins, which are responsible for many of the pediatric cancers, can serve as neoantigens and appear to be immunogenic. And a number of clinical trials are ongoing, testing different immunotherapies against these antigens. We also know that in some cases, high expression of PD-L1 in the tumor microenvironment, or if you see T cells infiltrating a tumor, that's a good sign. But here's the problem. The majority, 80% or greater, do not respond to these immune checkpoints, at least single agent or even when you give 2 of these agents together. So we still have a long way to go. So what have we learned about tumor mutation burden as a biomarker? Well, 2 people in my group a couple of years ago, Mark Yartrin and Alex Hopkins, wanted to better quantify the number of mutations that are a threshold beyond which patients will respond to these immune checkpoint therapies. And this, again, would allow you to go from a generalization to more personalized tumor treatment approach. And so what they had found was that, in fact, there's a threshold of 10 mutations per megabase that tells you those tumors that will respond. And the way they did this was they took a look at all of the studies that were published that showed responses to anti-PD-1, and that's what you're seeing here. And these were phase 2 and phase 3, and they did objective response rate on the y-axis and median number of coding somatic mutations on the x-axis. And interestingly, this is now an approved biomarker for at least Keytruda, the first study that was done to prove that this particular quantification was important. So, but as we know, there are some outliers, and I mentioned that there are different kinds of tumor antigens. So Merkel cell, as you can see here, clearly has a high objective response rate. What's interesting about Merkel cell is that there are two forms. There's the form that is UV induced that does not have a viral association, and those have a very high mutation, and they respond because of the mutation rate. And then you have those that are virus positive, and they have very few mutations, but they're very immunogenic because they are a virus. And then you have renal cell, another example of a high objective response rate with few mutations. And again, renal cell is the situation where we see these indels. And so what are the challenges then? So we know that there are a number of tumors that do respond, but many cancers don't. And so I've identified a number of challenges that we need to address, particularly in pancreatic cancer. We know that pancreatic cancer has a very difficult tumor microenvironment. There are a number of different signals that provide resistance to different forms of immunotherapy, and mostly these signals turn off T cells and in some cases B cells. We also know that there's significant heterogeneity. In the case of pancreatic cancer, the heterogeneity may not be as much at the genetic level as it is at the tumor microenvironment subcellular populations, including the stromal population. And then we also know that many of these cancers, including pancreatic cancer, never induce a good quality T cell. So even if T cells are infiltrating, they're not able to be activated to become anti-cancer agents. So the solution really is to better understand the signals in the tumor microenvironment and to be able to hit many of these signals at once in order to be able to get the best T cell response infiltrating the cancer. So as I pointed out in the start, I really believe that because we're in a technological revolution, we're now able to maintain the immunotherapy momentum. And this is going to be maintained because we're able to use these new technologies to answer key biologic questions in those cancers that don't naturally respond to our immunotherapies. We can also identify these new targets. I mentioned the indels, I mentioned fusion proteins. These are all now readily identifiable. Even 5 years ago, we didn't have the algorithms to easily identify these new antigens. But we can also identify new targets using single cell analysis, new targets for new immunotherapies. These are the signals that may be downregulating T cells. And also these new technologies hopefully will lead us to new biomarkers that can help predict better responses to our immunotherapies. So this is a pancreatic cancer, just a depiction. It's a human pancreatic cancer. And what's interesting about it is that although it has these islands of tumor cells make up at best about 10 to 40% of all of the cells within that tumor microenvironment. The rest is a stromal reaction. And we used to think that the stroma was inert, but it really isn't. In fact, the stroma is made up of many different forms of stromal cells. In addition, we also know that there are a lot of inflammatory cells that sit in the stroma. And these inflammatory cells in the form of macrophages, B cells, regulatory T cells, and immature myeloid cells, dendritic cells, they actually provide inhibitory signals interacting with the tumor, with the fibroblastic stromal cells to exclude T cells from the environment. So what we're trying to do in pancreatic cancer, and this again is the pancreatic cancer, this is a high, a microsatellite instability high tumor. This is a colon cancer tumor. We're trying to turn this pancreatic cancer, which has very few T cells, but mostly regulatory cells, into one that looks like a colon cancer that becomes now susceptible to immune checkpoint therapy. So basically what you're seeing here is CD8 T cells infiltrating around the tumor. Once you give anti-PD-1, you activate those cells and they can infiltrate and they can cause killing of the tumor. And so we've been testing the hypothesis that it requires at least 2 steps to reprogram the tumor microenvironment and optimize immunotherapy. The first, of course, would be to induce a good T cell. We've been working with vaccines, and I'll talk a little bit about that and show you some of our data. These vaccines could be shared antigens. That was the older form of vaccines we were looking at. We're also looking at neoantigen targeted vaccines. Many people at Dana-Farber and other places are also looking at neoantigen targeted vaccines now. That's something that we can do because of technologies that we have. But in addition to that, we also, once you get a good T cell induced, you want to prevent it from being deactivated once it gets into the tumor. And that occurs by giving checkpoint agents that block the inhibitory signals. These are some of those signals. We need to better understand in pancreatic cancer which of these signals are most important. And it may be a dynamic process. When you inhibit one, others may now compensate. So we have to keep in mind that the tumor microenvironment is a very dynamic environment. In addition, what we're finding, and I'll show you some data, is that agonist signals are very important in fully activating the best T cells. And we haven't really looked a lot at these agonist signals yet, but they're starting to make it into the clinics. My biggest concern, to be honest with you, is that because agonist signals alone typically are not enough to really make an impact in cancer, many of the ones being developed often are kind of put on the shelf because as single agents, they haven't worked. So we've been developing a platform study in the neoadjuvant setting of pancreatic cancer. And we find this to be a very important setting for being able to get a lot of tissue to study what's happening to our therapies. And the way we started is we started quite a few years ago now. Our first publication was in 2014, where we were giving our whole tumor cell. It was 2 allogeneic lines expressing GM-CSF 2 weeks before surgery. We would get the tumor, give the vaccine, and then at the time of surgery, we would be able to get the tumor and take a look and see what's going on. And then we would continue in the adjuvant setting. And this was our team, Lei Zhang, who really helped to develop this platform, Chris Wolfgang, our surgeon, Dan Lahiro, our medical oncologist, and Eric Lutz, who is an immunologist with us at the time. And we did show that we had significant improvement in disease-free and overall survival. And we also showed an association with high avidity T cells that were induced against one of the shared antigens, mesothelin. But here's what we really saw, and this is what we've been following up on. We saw these lymphoid aggregates coming into the tumor. So this is a pancreatic tumor just 2 weeks after one vaccine, and you could see these unbelievable lymphoid aggregates all through the tumor. And we see this in about 75 to 80% of patients when we just treat with vaccine alone. And when you start to study these, what they are there areas of or sites of lymphoid activation and potentially regulation, because we go from a antigen naive T cell to one that's experienced. We see markers of T cell trafficking into the tumor. We see evidence of early activation. We see CD4s, we see B cells. So they really look like lymph nodes sitting in the tumor, again, sites of activation of a T cell. But what we don't see are the lytic ability in these lymphoid aggregates yet. So most of these lymphoid aggregates just look like they're activating a T cell. And in fact, what happens is when you get these T cells coming in, we start to see regulatory signals going up, such as anti-PD-L1. And of course, that happens because these T cells are making gamma interferon. And you could see this in the lymphoid aggregates. And in fact, we had shown at that time, based on the immunohistochemistry we had at that time, that these lymphoid aggregates had these monocytes and dendritic cells that were upregulating the PD-L1 in addition to the tumor cells over here. And so working with Lisa Cousins and Takahiro Tsujikawa at the time, and this was through our Stand Up to Cancer grant, we were able to develop the first iteration of our multiplex immunohistochemistry approach. And basically we developed 3 panels of 12 markers each. And what was interesting about this particular approach is on one slide, we can stain up to 8 markers. And that's done basically by staining one. We get a picture of it, we digitize it, get a picture, and then we strip it, and then we just keep doing it. And you can do it for 12 times before you lose tissue integrity. And of course, you could start to do densities, heat maps, and you could quantitate. And we were able to show that in patients who had this low myeloid and high CD8 ratio, these were the patients who went on to long-term disease-free survival and overall survival. I'm sorry. But if you have a high myeloid, you are less likely to do well. And in fact, unfortunately, you would die pretty quickly. Also, the ratio of T helper 1 to T helper 2 is important. So having a higher T helper 1, T helper 1s help to enhance the CD8 cytotoxic activity. We also wanted to see which of the cells were expressing PD-L1 and what did it mean as a biomarker. And so what we found at the time was that, in fact, and we compared this to neoadjuvant chemotherapy versus vaccine. The vaccine is blue, the clear or white is the chemotherapy. And what you're seeing is post-vaccination changes. And you could see that the more mature macrophages and dendritic cells have larger expression of PD-L1. And in fact, we found that if you had higher expression of PD-L1 with the vaccine, but not with chemotherapy, you ended up going on to doing well, which of course led us, I'll pass that in, led us to the concept that now we should be adding anti-PD-1 to our vaccine. And in fact, not only have we added anti-PD-1, but this is, as I mentioned, a platform study. So now we have our third arm, which is vaccine, anti-PD-1, and anti-CD137. And this is based on accumulative data over the past 5 years, where our vaccine brings in antigen presenting cells, they activate the T cell, you get T cell infiltration, we get upregulation of PD-1 and PD-L1 on the tumor cells and on the macrophages. We also get PD-1 on the T cells. So this is a situation where we could be down-regulating the T cell. We also find that CD137 also goes up, an agonist signal. And so that's why we decided to add CD137 to the third arm. And the hope is that by adding all three, we'll get the best activated immune response. And so these are our three arms. We finished the chemotherapy arm I showed you. We just finished the anti-PD-1 and vaccine arm, and we're just finishing up the anti-CD137, anti-PD-1 and vaccine arm. And in fact, for the first time, we're seeing in the third arm, 3 of 10 patients so far demonstrating just after one triple treatment dose, we're seeing partial pathologic responses at the tumor prior to surgery. So this is the first time we've seen that. And so we're extending this arm to see if we could get a better approximation of the number of patients who might have this type of response to the triple combination. So of course, this is an opportunity as well to do single cell T cell receptor sequencing and transcriptomics. And so again, this is all data now that's ongoing. We haven't published yet, but what I'm showing you here are some of the studies that have been published that have really helped in single cell analysis to better understand the biology of these T cells to PD-1 therapy. And so there's been a number of studies listed here that have shown that, in fact, it's not the exhausted T cells that respond best to PD-1 therapy in the case of tumors like melanoma and lung cancer, which to me was kind of surprising. But in fact, they're bringing in new T cells to better activate those new T cells. And it makes sense when you think about it, because the neoantigens, they have a high neoantigen burden that keeps turning over. So most likely it's a dynamic process with new T cells coming in. And when you give anti-PD-1, those are the T cells most likely to respond and probably the most fit to respond to anti-PD-1 therapy. This was also shown that in basal cell and squamous cell carcinomas, that actually not only are you inducing new T cells that can be activated, but the number of exhausted cells go up too, because with PD-1 therapy, you do eventually get exhausted cells. So I thought that was interesting too. So of course, with just PD-1 therapy alone, we do know that eventually T cells can become exhausted. And again, you're generating new exhausted T cells shown at a single cell analysis level. And then also very interesting, I thought, and I'll show you why with our data, that a high ratio of PD-1 positive CD8 T cells to PD-1 positive Tregs until correlated with best responses in non-small cell lung cancer and gastric cancer to anti-PD-1 therapy. And this is important because, again, we used to think that PD-1 upregulation was associated with early exhaustion, but it may be that PD-1 upregulation without other exhaustion markers actually means you have an activated T cell. And so Luciani in our group has been doing the single cell analysis with this particular clinical trial I mentioned. And what we're able to do, of course, is annotate into different subtypes with single cell RNA-seq. And then we can actually generate through computational analysis a cell state trajectory going from inactivated to active to very activated or exhausted. And we can integrate this transcriptomics data with TCR sequencing as well. So I will show you that. And so this is what we've been able to do. This is a UMAP plot of first the CD8 T cells. We're working on the other subtypes, but you could see that we have multiple populations, the naive, early activation, some activated T cells. And we believe the CD8 granzyme B PD-1 population is the most activated and correlates best with the patients who are having this partial response at their tumor. And this is just looking at our violin plots, and we're looking at pseudotime. So basically, for those of you who haven't worked with pseudotime, the computational analysis using pseudotime gives you basically the ability to look at different, it's a snapshot of the different stages of the T cells at one time point, and that's what we're looking at. So these are the CD8 T cells that are naive, those that get activated, and you're looking at how much of them are activated. We have more that are granzyme B and more that are very activated. This isn't showing you which arm is doing it yet, it's just showing you what we're seeing at the single cell level. And now you get to the different arms, and vaccine alone is this arm here, the dark blue or purple. The pinkish one is vaccine plus anti-PD-1, and then the yellow is going to be our anti-PD-1 and anti-CD137. And you could see that it's the yellow, so it's our triple combination that gives us the most granzyme B, PD-1, CD8, as well as CD8 granzyme B. And in fact, when you look at the violin plots, you could see the difference here by vaccine group. So vaccine alone, vaccine plus anti-PD-1, and then vaccine plus anti-PD-1, anti-CD137. So clearly the triple therapy seems to be the one we want to continue and figure out, are there other signals? Because only about, in a small study, 30% seem to have the partial response after one dose. We don't know what happens long-term yet, but we're very optimistic that we will learn more from this study. And of course, we have lots of signals to look at. This shows you the heat map. You could see vaccine alone, not a lot of heat going on there. Vaccine plus anti-PD-1, we have a lot of signals that are elevated, but obviously the most signals that are elevated are in the triple combination. And lots of data, as all of you who work with big data know, it takes a while to really analyze this data, but we're getting there. But looking at the TCR-seq and integrating it with the transcriptomics is also very interesting for us. So basically, what you're looking at here, the light green is the granzyme B PD-1, the dark green is granzyme B without PD-1, and the dark blue is CD8 naive, and then this kind of in between is kind of an early activation. And basically the yellow is our arm C, and those are individual patients along the x-axis, and you could see that the ones in yellow are the triple combination. There's the ones that have the most increase in number of clones. When you look by treatment type, vaccine alone, vaccine plus anti-PD-1 is B, and C is vaccine plus anti-PD-1 plus anti-CD137. We have the most expansion of T cell clones that are, of course, expressing the most activated T cell markers. So ongoing, of course, we're analyzing other markers of response in the CD8 T cells to further establish the range of cell states. We're also analyzing some of the other subsets. We have all the data, it just takes time to analyze. And we've now moved to spatial transcriptomics, and that's ongoing. And I'll show you a little bit of that data. Again, none of this is published at this point in the pancreas cancer study, but we're working on it. And so Alana Fertig, who is my partner working she's a computational scientist. She does the data science part of what we do. And she developed something called COGABS, Coordinated Gene Activity and Pattern Recognition. And basically what this is she looks at the different gene expression, and then she looks at the amplitude, and by combining the two mathematically, she has developed an algorithm that can allow her to pick out patterns, and not just patterns in the data itself, but she can apply this to integrate different data sets, and she can apply this to the spatial transcriptomics. So that's what I'm going to show you. So we're working with 10x Visium. We're part of a small consortium to develop this on FFPE samples. And so here you're looking at what we've developed. We started out with breast cancer. It was a little easier than pancreatic cancer. As most of you know, transcriptomics and pancreatic cancer takes a little bit more tweaking. But basically what you're looking at here is our ability to apply cogaps to different areas within a biopsy of a breast cancer. And so the red is the invasive breast cancer, the yellow is the DCIS, so the pre-malignancy, and the blue are the immune markers or the immune cells. And then what she can do is look at patterns of transcriptomics within these and then develop these different patterns. And we could start to look at what is being expressed by the different cell types and how close they are to each other. So that is ongoing. And we've been able to do this with our lymphoid aggregates. We're getting there at least. This is early. I don't have a lot of data. Hopefully we will have more data soon and be able to publish, but taking a look at those lymphoid aggregates, you could see looking at B cell markers, T cell markers, where they are, CD14 and CD1C, and we could start to look at what's being expressed within these little dots. And so that's ongoing, being analyzed, and hopefully we'll have more on that soon. So that's on the neoadjuvant platform. We also have a metastatic platform study where we're learning a lot. We get serial biopsies on these studies, and we're building, again, using a lot of these technologies to better understand what's going on. And as many of you know, T cells have multiple signals. They have all the inhibitory signals. They have lots of activating signals. I talked about trying to hit the CD137 agonist. I'm going to talk about not only anti-PD-1 as an antagonist signal, but also CTLA-4. In pancreatic cancer, we're finding that CTLA-4 is a very important target that we need to also engage, even at low doses, to get the best response. And so this is a lot of work being done by Young Lee, and she started out way back when anti-CTLA-4 was the first immune checkpoint available. We compared anti-CTLA-4 alone, ipilimumab, to our vaccine plus anti-CTLA-4 ipilimumab, and found that even in patients who were metastatic, failed multiple chemotherapies, we were able to get a third of the patients to survive 12 months or longer. And certainly that was interesting. Obviously, you know, it's just a small study. It doesn't mean as much, but what it did tell us was what was happening on CAT scan. And this was very important because what we were learning already from the study was that if you take baseline CAT scan, you could see these are the lung lesions. You could see that at baseline, they were pretty large. By 7 weeks, almost 2 months later, they were increasing in size. You could see here. So that was telling us that vaccine plus ipilimumab alone was, you know, it didn't seem to be working yet. If the patients waited 3 months, we started seeing the regression. So it told us that even pancreatic cancer patients could respond, but it takes a lot longer, particularly with this particular combination. And so we wanted to better understand what was happening. And so our team, led by Wan Jin Ho, who was a postdoc at the time, and Annie Wu, an MD PhD student, and Alana Fertig, we used mass cytometry to look at peripheral blood and also the lymphocytes within the TIL to see if we could better understand. And we were able to analyze both pre-treatment and at the time the patients were progressing on CAT scan to see what was happening with those immune cells. And it's an interesting story because we found something we weren't expecting, that yes, there's a heterogeneous T cell alteration or modification with these therapies, but it turns out that all of the patients have activation as well as evidence of inhibition. And so there was no difference between those patients. Half the patients went on to having evidence, radiographic evidence of response at 14 weeks. Half did not. We did not see a difference between what the T cells were telling us. And so I'll show you what that data looks like. What you're seeing here are These are radar plots, and these are all the CD4 subtypes on the left, CD8 subtypes that start at the bottom and go towards the right. The red are individual, each line is an individual patient. The red are patients who did not respond to the therapy. The green are those that did. And it looks very similar. We're seeing, at least overall, an enhancement of a CD4 response, in particular an effector memory CD4 response at 7 weeks post-therapy during therapy compared to baseline. When you look at it, we see many of the effector memory cells, both the effector memory and T helper and CD8 effector memory cells, whether you responded or not, upregulating these markers of inhibition, so TIM3, LAG3, PD1. The distribution, of course, is going to be different. It's not surprising that each marker is distributed differently, but overall, you could see that many of the T cell populations, regardless of treatment, have it. Interestingly, there's also activation. So CD28 seems to be upregulated no matter whether you responded or not. Red is the non-responders, green the responders. And again, it's quite diffuse for CD28 as expected. And we also see granzyme B going up on the T cells, the CD8 T cells. We see proliferation on CD4s and CD8s. We see OX40, that's one of the agonist signals, and 4-1BB or CD137 upregulated. So it seems confusing. Again, IL-2, TNF-alpha, interferon gamma. At 7 weeks, all these T cells seem to be activated, but they're also starting to show evidence of regulation. So basically, what we hypothesize then is that when we give anti-CTLA-4 with our vaccine, we're getting an activation of the T cells, and they're upregulating these activation markers, but they're also upregulating regulatory markers to compensate. And it's sort of a balance. And sometimes the T cells went out and they become killer T cells and they hit the tumor and the tumors start to shrink, but in other cases they don't. So we think we're on to something, but we think we need to do a lot more to get a better response. So we've been moving this forward. And Young Lee just reported her data on our prime boost vaccine. So a second vaccine we're looking at, Listeria, expressing mesothelin, a shared antigen. So priming with our whole tumor vaccine, giving then a Listeria vaccine, which is also a good adjuvant, expressing a specific tumor antigen. And we compared the prime boost alone to that with anti-PD1. And interestingly, we have a similar finding. We just published the data on this. Pre-treatment, you could see, again, these are metastatic patients, failed chemotherapy. We have tumors here. Week 10, they're progressing. Week 30, this patient went into a complete remission or close to a complete remission radiographically. This is week 10, we got the biopsy. And in fact, on biopsy, we could see CD8 T cells coming in to this tumor at week 10 that wasn't there pre-treatment. And when we start to look with our multiplex immunohistochemistry, we're able to start to see what's going on. We see quite an increase. This is that one patient in CD4s, some increase in CD8s, yet this patient looks like they're progressing at this time. And we also see what appears to be reduced exhaustion and an increase in early effector memory cells. When we look at all the patients, what we're finding again is that if we see an increase in TAM at baseline, it doesn't matter how well we do with the T cell, those patients are just not going to do well. They'll have a shorter survival. Even if we reduce them with our immunotherapy, whereas if we have a lower TAM evaluation, those patients are the ones that have long-term survival. So these are the tumor-associated macrophages. Interestingly, and this is looking now at all the patients on the study that we had biopsies for, which is about 35 patients. Interestingly, we did not see a significant difference baseline at T cells infiltrating. That did not tell us much. There was a range of T cells, but certainly we wanted to see those T cells in patients who went on to long-term survival. And again, we saw reduced exhaustion and an increase in early effector memory T cells in those patients who went on to long-term survival. We also did sequential T cell receptor sequencing in these patients. So we went back to both the vaccine plus ipilimumab and compared that with vaccine plus anti-PD-1 to see if we see differences in TCR sequencing changes. And this was done by a graduate student at the time, Alex Wang. And what he had found was that when he looked at patients pre-treatment to post-treatment, he looked at CTLA-4 non-responders versus responders, PD-1 non-responders versus responders. What we found was that the number of expanded clones seemed to correlate with the CTLA-4 responders. So if you went from pre-treatment to during treatment and expansion of your clones at week 7, again, everything is at week 7 when people seem to be progressing. If you expanded your number of T cell clones, you did better with anti-CTLA-4. So what this is really saying, if you look up here, anti-CTLA-4, if you increase the diversity of T cells available for vaccine priming, you had longer survival than if you didn't. In contrast with anti-PD-1, if you increase the number of clones that were already antigen experienced, you did better with anti-PD-1. And actually, this seems to correlate now with what people are seeing with melanoma. And so we have an ongoing study where we're now randomizing patients between low-dose ipilimumab, 1 milligram per kilogram every 6 weeks with nivolumab, anti-PD-1, and our listeria vaccine versus our prime boost vaccine with the same ipilimumab and nivolumab, because what we're hoping is not only will we be able to increase the number of patients who respond, but we're hoping to increase how fast they respond, because imagine if you increase the number of T cells available for initial priming, maybe you'd have a faster response. So that's our hypothesis. And our hypothesis seems to be showing true in some of our patients so far. So in 8 of 20 patients that were treated so far, we're seeing that even in liver lesions that normally won't respond to anti-PD-1, we're seeing within 6 weeks regressions, and that correlates with their biomarker CD19 going down. You could see here. We see this with another patient with a liver lesion, again, just at 6 weeks. Remember that these were the patients who would have been progressing at that time. They seem to be responding, and even peritoneal metastasis, which we've never seen respond before, seems to be going away with IPI plus Nivo and our vaccine. And so currently we have this, as I mentioned, we have this platform study. We've just added a few other agents. Currently we're adding CXCR4 inhibitors to target as well. And that study just opened up to our platform study, and we're looking for our next agents. The goal being that we want to be able to reduce the TAMs, tumor-associated macrophages, and also try to reprogram the different subsets of cancer-associated fibroblasts. And so I'll conclude by saying, I think we're starting to see where we can push immunotherapy toward a good place for even cancers like pancreatic cancer. It certainly starts with improving antigen processing and presentation and getting an optimal T cell signaling. This may also be done with adoptive T cell therapies or CAR T cells, and there are many of those approaches that are being tried even in pancreatic cancer. That I'm aware of, and I hope that they will be successful. But I think we also need to be thinking about these agonist signals in addition to the inhibitory signals in order to make not only a vaccine-induced T cell more potent, but also probably the adoptive T cells as well. We need to reprogram the monocytes and fibroblasts. I think this is very important. Those TAMs, myeloid-derived suppressor cells, and the CAFs are clearly signaling to prevent T cell entry and T cell function, and they crosstalk with the tumor cells. And then, of course, I haven't even talked about some of the other interesting areas, including the microbiome, the vasculature, and of course, metabolomics, which is very important as well. So I think there's a lot more to do, but I think we're getting there. I think there are many examples of others who are showing some good progress with agents like anti-CD40 and with adoptive therapy. So I'll stop there and just also show you all of my scientific partners. I've mentioned the ones who have worked with me on the studies I presented and also the funding we've had. Thank you, and I'll take any questions.
Great. Well, thank you very much, Liz, for an awesome talk. I'll encourage anyone in the audience to submit questions through the question and answer. Maybe I'll start out by asking, I was really fascinated by the comment that you made about responses in the, not seeing responses in the liver or the peritoneum, but obviously you showed some nice responses in the lung. Can you comment a bit more on differences in the immunologic responses you've seen on the studies in the lung versus the liver? And obviously it's just a fascinating topic overall, because as you know, patients with lung only metastases often do better and have a bit of a more indolent disease. So it's fascinating to think about what the interaction with the immune system is there.
Yeah, Andy, that's such an important question. And Juan Junho in my group did do a study to try to address this, where he used mouse models with the same tumor type that responds to anti-PD-1 MC38, and he did a orthotopic liver metastasis versus lung metastasis. And we did single cell analysis and try to look at both the regional lymph nodes, draining lymph nodes, as well as the sites. And it's interesting, it's not a yes or no situation, but what you see in the lung is many more antigen presenting cells that are mature. And in the liver, what you see is a lot more LAG3. That seems to be the big signal that is highest in the liver. And as we're seeing, we have a study ongoing in HCC, hepatocellular carcinoma. Anti-LAG3 seems to certainly enhance the anti-PD-1 response, at least at the liver. So I think that LAG3 seems to be an important inhibitory signal in the liver. There's also obviously other signals, I'm sure there, but they're mostly quantitative from what we could tell. Qualitative, not yes or no type of signal in one versus the other. But there does seem, and it makes sense in the lung, I would think, because the lung gets all this bacteria coming all the time. So it's much more primed to be able to present antigen to T cells. But more needs to be done. That's just one example. We're very excited to look more into that.
Yeah, that sounds like a nice model system to study it. We have a couple of questions from the audience. Samantha Pereira asks, thank you for a great talk. What do you think would be the best way to increase mutational load without collateral damage?
That's a great question. The DNA damage agents, there's a lot of work in that area. So there are people trying to look at it from there. We're trying to look at it from using epigenetic modulation, so uncovering new neoantigens with epigenetic therapy. But I'm definitely interested if you have other suggestions too. And we're certainly, we have some clinical trials where we've seen some good responses with HDAC inhibitors and immunotherapy. So we're looking now to see if it has to do with changing the mutational load.
Excellent. And Sandra McAllister asks, are any of your patient cohorts large enough to evaluate whether there are age stratified responses to therapy in those trials?
That's a great question. And unfortunately, in these platform studies, it's more just trying to get at the biology. And that's an important question. But I do have to say, you're asking a really key question. And I'm collaborating with Ashani Ranta. I don't know if you know her. She's come to Hopkins, and we're co-mentoring a fellow, Dan Zabransky, who's been looking at fibroblasts, or the normal fibroblasts and the malignant fibroblasts in patients who are 50 plus versus patients who are under 50. And we're actually starting to see a difference in tumor aggressiveness based on that. So I think there's a lot to be done in that area. I think it's a really key question, but unfortunately our studies aren't large enough to really discern that yet.
Great. And maybe I'll just ask another question. Have you looked at sort of the basal-like versus classical subtypes and how they respond differently? I know there's certainly some correlations with the basal-like subtype and increased interferon responsiveness in the tumors, but I wonder if there are any immunologic correlates of that.
That's a really great question. We're currently looking at that in some 3D modeling to see if we can get at that question. So that's work that's being done by one of my postdocs right now, but we don't have any data yet. I think that's also a really key question. I've loved the work that's been done at your institution, looking at the differences. I think that's a really important advance.
Great. Edward Fritsch says, nice talk. How do you imagine anti-CTLA-4 is working to activate Tregs in your neoadjuvant setting compared with IPI monotherapy? Is this specific to the vaccination setting?
Yeah, so I think that, I've been pushing, particularly in pancreatic cancer, since, T cells are the problem and you want to have as many T cells available. And so I think low dose IPI, this is 1 milligram per kilogram. So it's lower than the 3 milligram dose that has been approved for melanoma treatment. And I'm recommending that this low dose, just even given at the time of initial immune priming, whether it's with chemotherapy or with vaccines or whatever you're doing, it's important to expand the naive T cells available for priming. And so it may also be altering the Tregs, and we are looking at that. We don't see a change in the number of Tregs, but what we are seeing is a change in the function of the Tregs in these specimens from the neoadjuvant study. And I think that goes along with what Jim Allison has said. In the mouse models, the anti-CTLA-4 antibody in the past has been a Treg depleting antibody. There now is one that's a inhibitory rather than depleting. But the human one, at least ipilimumab, is more of an inhibitory, and we do see evidence that we're altering that function. But I think the other interesting part is the TCR-seq data saying that we're increasing the number of T cells available for priming.
Great. I guess I'd also be interested to know, Liz, what your thoughts are on, RAS inhibition in pancreatic cancer. Obviously, there's some nice data from Amgen and other companies that have developed RAS inhibitors that these inhibitors are immunomodulatory, even beyond the inhibitory effect on the cancer cells themselves. And RAS is the holy grail in pancreatic cancer, and there are new drugs coming to the clinic in the next 3 to 5 years that will be useful for pancreatic cancer, the alleles we see in pancreatic cancer. How are you thinking about that, and what's the data maybe back to MEK inhibition or other RAS pathway inhibitors in immunotherapy?
Yeah, no, I think you're absolutely correct. KRAS, sometimes I show that slide of what mutated KRAS can do to the immune system. I should have shown it today. But yeah, it's really a problem. Mutated KRAS, causes IL-8 production, which, as we all know, alters the monocytes to become suppressive. It interacts with the stromal cells to become suppressive. That's just a start. It affects metabolism of T cells. I mean, it has so many effects. And I do think, I'm very interested. We were talking about that today. Actually, we had a little seminar here. We were talking about trying to look at that. The problem, as you know, is that the current inhibitors don't really hit the most common mutations in pancreatic cancer, but I'm certainly keeping my eye out for those that hopefully will do a better job of hitting our KRAS. We do have a KRAS targeted vaccine in the clinics. It's in the adjuvant setting where we are seeing, and this is being done by Neha Zaidi, who's a new faculty with our group. She's already showing in a handful of patients induction of T cell responses against multiple epitopes, including the one expressed by the patient's tumor. This is the adjuvant setting, so it's really a test of the safety and the immune induction, but we're actually going to the pre-malignancy setting with this vaccine pretty soon. We're already at the IRB for that with the hope that we can actually prevent progression, and it's based on some of the modeling we've done in the pre-malignancy where we've published showing that we can prevent early PANINs from progressing by targeting mutated KRAS with the vaccine. So that's another area I didn't show today, but I'm very excited about, and I think there's a lot of opportunity with the inhibitors to even do better.
Well, that's very exciting, given that low-grade PANINs with RAS mutations are common even in patients who don't go on to get pancreatic cancer. That's a huge opportunity. Sandra McAllister asks and acknowledges that delivery of STING agonists would be an issue, but says, do you have reason to think low-dose STING agonists could be beneficial with IPI to stimulate priming?
Absolutely. And in fact, we've published that preclinically. Yes, yes, yes. I think STING is important. I think it works a little differently in humans, but I'm very excited to do that. We were working with Aduro Biotech, and for full disclosure, on their STING, and then they've stopped that program. So we need to figure out how to move forward in the clinics with another group.
And maybe I'll just ask your thoughts, Liz, since you're so involved in leadership of some of these large consortia and at a national level. How do you envision some of these efforts like Breakthrough Cancer or the Parker Institute and working together across institutions? Do you see barriers or opportunities there that we're not taking advantage of as a community?
Yeah, I think, just as the question of, can I really interpret, whether older people do better than younger people or vice versa. I think, if we just don't have enough data, to get data on, a handful of patients in each clinical trial, even 30 patients is never enough, because we know that we don't get serial biopsies in everybody to begin with. If we could have a consortium group that is able to not only, contribute technologically to understanding these treatments through serial biopsies and other biospecimens, but also to be able to share data, I think we would make great progress in this disease quickly. And in fact, I think the COVID situation tells us that when you share information rapidly before you, and it's hard, don't get me wrong, I understand the challenges, particularly for early career faculty, but we have to have some system, and I feel very strongly about this, where we protect our young faculty and of course trainees, allow them to publish, allow them to get the accolades for what they do, but also be able to do it in a team science way. Well, that's the only way we're going to make progress. And I think that's where you're headed, Andy. We really need to do this. And I'm hopeful that these groups, PICE, Parker Institute, Breakthrough Cancer, Les Garden Foundation for Pancreatic Cancer, I mean, this should be the goal. This should really be the goal.
Yeah, I think your platform trials are a nice template where those could work similarly across institutions and sharing data in a similar manner. So Samantha Pereira asks, again, to follow up with the STING question, what are your thoughts on why the clinical data is divergent from preclinical data? And then thank you with 2 exclamations.
Yeah, that's a good question. Again, clearly STING works differently in the preclinical data. I think the innate immune system's different. So even the toll-like receptors are different if you think about it between the preclinical and the clinical. So we really need to get a better handle on what those differences are, but there's certainly different signaling, so different molecules signal differently. There are different compensation mechanisms as well, I believe, in the innate response that makes it different. So I think those are some of the differences. And then I think that, the inbred mice, are going to be very different. They're not exposed to infection. So the diversity, again, of signaling is going to be different in the mice versus humans. So there's a lot. So the microbiome, obviously, with the innate immune response. I think the innate immune response is probably more affected even by the microbiome and other components than the adaptive immune response. So I think that's where we need to look for those differences.
Well, great. Well, I think we're at the hour. If we have no further questions, I'll just thank Dr. Jaffe for a terrific talk, really outstanding work. And thank you for your leadership in the field as well. And thank you to the Krakoff family for their generosity in sponsoring this lecture series and for all they've done for Dana-Farber over the years. So
thank you for inviting me, and thank you all. I appreciate it.
Thank you very much.
Bye-bye.
Bye, everyone.